Right. So, hello everyone to a very special episode. Uh, I am an unfamiliar voice. Probably you may have familiarized me from a couple of other episodes, but I am not Sahir Hussain. I am, in fact, the brother of, and we have a special guest, Sahir Hussain, who you may be acquainted from a particular podcast, maybe this one. Uh, and
You'd hope so. You'd hope so.
You'd hope so. And with me co-hosting is Jeremy. Frequent listeners may be well acquainted with him more than myself. So the host, well, will now ask the guest to introduce himself. Sahir, take it away.
Oh dear. Yeah, on the good note, this is what, episode 48. So we've done a lot of episodes before we've come down to this, where we're talking about my work, but we finally got here. And I think you've done a very good job with the introduction, in the sense that I think people should know you more than Jeremy, because Jeremy's only been on one episode. He's done a lot of behind the scenes
Ah,
stuff.
I
He's
see.
done the logo for the show and a lot of the social media stuff, but he hasn't been on, except for that one episode. So yeah. Anyway, thanks guys. Glad to be on the other side. Now I know how it feels when to be asked questions as opposed to asking. So yeah, excited.
So let me start off by giving the viewers a little overview of what your accomplishments are, to my understanding. So you you did your bachelor's in psychology and international relations, uh, f- following which you did your master's at King's College London in psychology and behavioral neuroscience. Would that be accurate?
Just
After which,
neuroscience.
just neuroscience. I apologize. That just tells you how close we are as brothers. Um, and and then came back to your stomping grounds at the Victoria University of Wellington to do your PhD under your beloved supervisor Bart Ellenbrook, who has previously appeared on a couple of episodes on this podcast. Uh, and and could you please tell us a little bit more about your journey and how you got into psychology?
As a family member, you know the journey. But for Jeremy's sake,
Yeah, for me.
uh,
Apparently, not as accurately as I should know.
it's it's fine. I still tell people that you're doing something with AI, although yeah, I just say I ask my brother if I need computer problems fixed. That's it. So that's how much I know about your work.
Yeah, I got a
All right.
screwdriver with me.
All right. So yeah, for Jeremy and for the audience, so the way the story goes is we did high school in India. So we were doing schooling in India, and over there the rule is that you either become a doctor, an engineer, a lawyer, or a disappointment to your family. And uh, for one reason or the other, I didn't do any of them. But I did want to come back to New Zealand to go to university, and I had gotten some really good marks in environmental sciences during my final board exam. So the board exam is like your final year 12 examination, which you have to do, which is compulsory. And one of the compulsory subjects at the time was environmental sciences, and I'd done really well in that. So I had applied to Victoria University to do environmental sciences. Um, at the same time, mom and dad were like, you can't just sit around at home for 6 months until, because we had finished in June and university here starts in February. Mom and dad said, you can't sit around for 6 months doing absolutely nothing. So they told me, go to a university here for those 6 months, do something. And I picked up psychology there, just to kill time, psychology and marketing. And English literature, psychology, marketing, English literature. That was the subject thing. And I ended up, instead of, I think I went to 6 classes across the 6 months. It was good. I had a lot of time, spent a lot of time playing basketball and cricket. But yeah, I did actually enjoy the psychology class. So then I applied. When I finally got into uni, I started off doing a double major in psychology and environmental sciences. And then, as Amer mentioned, Bart Ellenbrook. So Bart Ellenbrook uh, is a researcher at Victoria University of Wellington. He works on drugs mainly. So he does drug research on psychedelics, nicotine, alcohol, which I will talk about, and so on. And he was a behavioral neuroscientist, and what he did, some of his lectures in first year were the most interesting, and that sort of made me veer in that direction a bit more. The environmental sciences lectures were a bit boring, but the human geography side lectures were really cool, so I veered into that direction, and I ended up with my undergraduate degree in development studies and psychology, and then from there moved into neuroscience, and from there moved further into neuroscience with the PhD and so on. At every opportunity I tried to get out of academia, I applied for multiple jobs in multiple places around the world and got rejected from each and every one of them. So academia kept pulling me back in, and yeah, that's how I ended up with my PhD. Uh, yeah, that's the basic story. Jeremy, any thoughts, qualms, queries?
Many qualms. No, I mean, like, for someone that's known you for this long, probably 7 or 8 years now, it's nice to get a formal background on those things. And I know a little bit about what you've been studying and what you've been doing. So I guess we should delve into that a little bit. My awareness of what you've been doing revolves around what you've told us in our like friend group chats, and that has always been to do with making like rats drink alcohol. And I know a little bit more about that, but I guess let's start with like, why are you making rats drink alcohol?
uh, yeah. So why? I know it's a good question if you think about it. If I offered an 18-year-old college student alcohol for free, I think they would drink it pretty happily. To be fair, in this economy, you offer anybody free alcohol, they will probably drink it. Um, but but the real reason why we do animal studies in general, so why I'm giving alcohol to animals, is because we're trying to isolate the effects of alcohol. And that's true for any drugs, any medication. The reason why we do animal research is generally because we're trying to specify certain conditions and isolate and look at the effects of a drug or an effect of any sort of behavior paradigm or whatever we want to look at, and just look at those effects. And that's why that's why we would use an animal model. So in this case, when I started off my animal research work, the first project which I was on was actually looking at um, MDMA and cocaine, and looking at the behavioral effects they had on rats with a certain genetic deletion in them. So the other thing about animal research is you can actively activate or delete genes in animals and see what that does. So the one which we were looking at was serotonin. So serotonin, people might know, I think it's come up in a few episodes, but it's around what serotonin does have a lot of work to do in the brain in terms of your pleasure cells. So in terms of like but it also has a little bit of role to do in motor coordination. So that's your movement coordination and things as well. So um, when and serotonin is basically controlled by your genes, right? So there's multiple genes for serotonin. The gene which we were looking at is called the serotonin transporter gene. All you need to know is it kind of controls or regulates the amount of serotonin in the brain and in the body. And these rats, half of them did not have that gene, so therefore their regulation is different to like an animal which does. And you kind of see this in humans as well. In humans, there's different types of that gene. So the same transporter gene is of different lengths and therefore has different activity in different human beings. So we're trying to mimic that, and we're trying to see whether like a shorter serotonin gene, which produces sort of a shorter serotonin knockout gene, which sort of regulates serotonin to a lesser extent, whether that has an impact. This is not to say that it is the only gene, it's just the one gene we were looking at. And we were just trying to see how that gene sort of impacts behavior while taking the drug. So that's how we got into it. That's why we do animal work. And as you said, with my work, what my current work is, or my current PhD and research was around alcohol. So the first project which I did was looking at serotonin. those serotonin rats. So they were three types: the ones which had the full gene, the ones which had sort of half active gene or half functioning gene, and then the ones which had no no gene, or a knockout, the genes knocked out. And gave them alcohol and basically saw whether it sort of changed the drinking behaviors or whether the drinking behavior was different. So that was my first project, and then I can talk about my current, or like not my current, but my PhD project and my research after that later. Hopefully that
I'm
answers.
I'm curious based on that, and I'm sure Amir's got some probably a little bit more intelligent, a little bit more scientific questions to dive into in a second. But
Marginal.
I marginally, I'm curious from as a self-described empath. I'm curious what got you into the field of like looking at alcohol and drugs and their impacts. Like, is there a personal connection with those things? And if you had some kind of personal connection, whether that be emotional or like a personal experience, how did your research sort of change that connection and the way you looked at those substances afterwards?
Yeah, that's a really good question. So there's levels to this. So there is a little bit of a personal connection in the sense that when you grow up in an Indian household, usually mental health is like pushed under the rug a little bit. And sort of if you have family members with a sort of mental health problem of any sort, you kind of people just kind of like ignore them, being like, oh, they're a bit crazy. Let's just avoid them. Um, they're not necessarily say they're crazy, but they're just a bit weird. They're a bit off. Um, yeah. And so to that regard, that's kind of like that's from where like my interest in psychology sort of started, in the sense that I remember telling my parents, telling my auntie actually, I told one of my aunts when she asked, why do you want to do psychology? And I was like, to figure out what's wrong with you. Um, and And it's not to say there was anything actually wrong with her. It was just that I thought my family was crazy, like everybody thinks their family is. Um, but the more you sort of like study psychology, then the more you sort of kind of like understand like the effect of the environment on people. Then you do neuroscience and you do psychiatric genetics and so on, and then you understand the influence of genetics. Mine and Amer's mom's a geneticist, so we had that growing up, like knowing the entire role of genetics, particularly in like medical conditions, but then you sort of learn and grow that also your mental health and mental conditions also have that linkage. And yeah, the way I got into drug research is I think I found it the most interesting. There's a story which I think mom said on her podcast episode when we had interviewed her, Dr. Kurtulana-san. She spoke about genetics. That was really cool. But she said the story about Amer actually, where
Hey,
she
she
talked
didn't
about.
name names in her episode. Why are you snitching now? That's fine. Carry on.
But we were talking about codeine. So codeine's a drug which is just generally used in medication as sort of like in pain medication, right? But there's certain people in this world who have a genetic abnormality, not abnormality, abnormality is the wrong word, sorry, genetic mutation, where it sort of hyper metabolizes codeine, so it can be toxic to them. Uh, and you start hallucinating and so on and so forth. And Amer has that. And Amer was given codeine once because he got hurt. We won't say who hurt him. That can be separate. But he was given codeine and he started hallucinating. And that sort of idea that the same drug can affect two humans completely differently, and one can find like pleasure in it as it's a painkiller, but the other one can have horrific hallucinations for the same thing, sort of got me really interested in like medical drugs. But then that idea sort of carried over to alcohol, and then when you're 18, 19 in New Zealand and everybody's drinking or doing whatever, you sort of see like some people have like a much higher tolerance to one thing while the others don't. And that kind of got me interested in the sort of sort of that metabolic side of how drugs work and that side of thing. And then combining it to the earlier story which I said about mental health, and there are reasons why certain people might become addicts, although everybody uses or everybody not everybody uses, but a lot of people use substances. for various reasons, but only a very small percentage actually turn out being substance users of some sort. And kind of like, yeah, that's how I got my interest. It comes twofold, and yeah, that's basically the story of it. Did I actually answer the question? I have no clue.
Yeah, you answered the first part. I'd say the second part was, has your relationship with those things changed since then? So like specifically, I guess alcohol is probably the more common thing to
Yes.
consume.
Yeah. Yeah. Actually, it has a little bit. I think more than my relationship. I think the more political side of like how we should treat people who have problems with it has changed. Like you grow up and you think that they're bad people or whatever, and I think that changed a lot when you start working with drugs, knowing how they impact, well, animals in a controlled environment. But we're all doing this to learn for what we can do in public health, right? At the end of the day, all this research, all this work is to help people the best we can. And yeah, I think that's where it's changed the most. I think because the other things of, the signs that you see, don't do drugs, or this is bad, that's bad. That's true. Like that's not necessarily wrong, but I think the way we handle it, or according to my perceptions, changed a lot since yeah, the early days of, when you're 14, 15, 16, trying to sneak alcohol around, hiding it from your parents, to now. Yeah.
Nice.
So yeah, so carrying on. So let's delve a little bit more into your PhD research. To my understanding, it focused particularly on the effects which the male's drinking has on the progeny. Why did you want to look specifically in that aspect? We all know that people are well aware that pregnant women should not drink alcohol. So what made you try and investigate the father's influence in father's drinking in the progeny?
Oh, because I'm a feminist.
Nice.
Short answer.
um, no, you kind of answered the question to yourself there to an extent, right? So if I ask both of you, um, have you ever come across any information in your entire lives about sort of male drinking or men drinking in general, like in terms of public health, in terms of anything? Have you guys ever actually come across some serious information apart from, generally speaking, the general sort of things of like drinking bad or be careful? Like there's been nothing specific, right?
Yeah, no, nothing. Aside from mental health stuff that's cropped up in the last maybe like 5-10 years.
Exactly. So yeah, apart from, yeah, for your mental health a little bit, things like that, but generally uh it isn't brought up. And one of the reasons it isn't brought up is because drinking is a part of so many cultures, right? Like um psychology is in a sense a Western science. It comes from the West. The idea comes from the West, and in most Western countries, drinking is accepted, encouraged, all these things, to some extent. Of course, again, when we get to the alcohol abuse side of things, again, there's taboos around it. But generally speaking, 80 to 90% of people in the UK, in New Zealand, in America, all have had some experience with alcohol. But if 80 to 90% of people have this experience, and the only sort of regulation which we have about it is advice to women who are pregnant, then either it's sort of like we're willfully ignoring it because we know that at an extreme it's bad, or we or we sort of haven't really looked into it. It's one or the other, right? Either there's been research and we've just like pushed it aside, or we haven't researched it at all. In either case, there's no sort of public health information about it. And the actual project, just because Amer said progeny and all that, I will just tell the title of my PhD and what it was. So, and my subsequent work for a year after that, what it was looking at how alcohol drinking in males, so in fathers, before conception, so before in rats, before mating or before having sex, before conception, how alcohol then affects the next generation. So again, there has been social research, a lot of social psychology research, a lot of social science research, a lot of sociology research on like growing up with alcoholic fathers or fathers who abuse alcohol. So there's a lot of things around that, right? It's very simple. Don't be drunk in front of your kids. Don't mess up your kids by beating them up. I think we've all kind of. I think it's generally known now. Whether people practice it or not is a different story. But I think that part's generally known. But there's very little about like sort of alcohol in terms of while trying to have kids, how alcohol might impact the children or like the development of the child. There is a little bit of work on sperm count. So alcohol can reduce sperm count, and sperm mobility, and there's a little bit of advice on that in terms of men with low sperm count. They sort of get advice of like, reduce your drinking, that will help increase sperm count and things. But there's not a lot of work on sort of how on how alcohol can impact like the sperm in such a way that conception and development is affected. Now, I say all that to say that the reason why this is important is because 50% of all your genes come from your dad, right? So like 50% come from the mother, 50% come from the father, and it's not equal. That's one thing which needs to be clear, in the sense that you do not get half from the mom, half from the dad, and then you're a mix, in the sense that you get your body just decides which ones will be active and which ones won't. No, there's certain genes which only always come from the father, and there's certain genes which always come from the mother. And a lot of the genes which come from the father are actually very important in early development. So one good example is this gene called IGF-2. It's called insulin growth factor 2. And it is very important for the early growth of the baby, fetus, whatever you want to look at it, particularly in the womb. And there is a maternal gene, or in the gene in the mother, called H19. Now, the job of H19, in essence, there's a lot more, is to sort of control this growth of the fetus. Because if the baby is too big in the womb, then there's birthing problems. So you don't want the baby to just keep growing indefinitely. But if the IGF-2 gene from the father isn't working, then the baby will be small, right? And that's, again, evolutionarily not what you want. You want a healthy sized baby. And so there's these genes which sort of play together, and alcohol tends to impact this gene. There's evidence to show, at least, that alcohol sort of impacts this gene, among many. I'm just using one example here. And if alcohol can affect this gene in the fathers, then what you would see is that the baby might be smaller because it's not growing as much, and the H19 gene is overpowering from the mother's, like sort of overpowering and suppressing the child's growth a bit more, and then you get babies which are like underweight at the one end, or at the lowest end might not have developed completely and might lead to other complications at the furthest other extreme. Of course, I need to say that all this research is relatively new, in the sense most research into this field has started. Most of the work started from 2014 onwards. So there has been work done before, of course, but most of it generally in this domain, most of the papers that you see would be around from the 2010 to 2020 sort of area. So it's all new. A lot of work still hasn't done in humans. There's so many other factors which you need to look at, but that's why animal research is important, to try to see and sort of identify exactly what's going on, or exactly what we can expect. So then when we look at it in humans, we can sort of account for all the other factors like diet and other things, diet, physical exercise from the mom, physical exercise from the dad, so on. Yeah.
Right. So I've got two questions from this. So you first mentioned that there's not a lot of research conducted in this area, specifically due to the prevalence of drinking in most Western cultures. So the research you did find, were there any specific regions in the world where these papers originated from? And were they more catered towards drinking, or cultures which are very much conservative about drinking?
See, I think there's an issue here, but this might go to a wider issue in science, and wider issue in research. That most papers which I came across did come from either America, Japan, the UK, Europe. Netherlands does a lot of work on this. Germany does a lot of work on this, and a lot of the reason is that most big journals only publish from these countries. Like there's a bit of a bias there. Also, all these journals were in English, which helps me, right? Because I'm not capable of reading many other languages to an extent where I can derive, uh, yeah, key research from there. So there's a lot of bias involved in those things. So, but a lot of work generally, which is done, and especially in human populations, would be from Europe because they do a lot of, for example, Netherlands, Finland, all these countries with socialized healthcare systems have a lot of data as well, where they can, which they can use for these things. So yeah, most of the research would have been from, I would say, the same countries where alcohol is drunken, but and yeah, those countries also have the money to do the work, so that's also a big part. So yeah, I think all those factors play a role in what evidence I found.
So this would actually be a good little plug for one of your episodes, which discusses these aspects in academia, um, and journal publications, and it is a fascinating listen. So I would encourage listeners who haven't listened to that to go back to that episode. Um, but yeah, so the second question I had, since you did say that a lot of this work is very recent, is it fair to say that uh, the research conducted in this area does not yet have the capabilities to investigate the long-term effects?
Um, for some things, yes, and for others, no. Because it really depends what we're asking here, right? And I feel this will be my answer for a lot of questions about speculation, because that's how science works. You can tell some things, you can't tell others. I don't know. Because for example, again, countries which have pretty good health data, so Finland, Denmark, the UK, even the USA to some extent, they've got a lot of large cohort studies which have been going on since the 80s, 90s, whatever. All of them now have 2, 3 generations of people to look at, and they can probably track back depending on how good data information is. Now we have the technology with machine learning getting better, with statistics getting better, having more powerful computers. We have the ability to like look back and sort of decipher things which we previously didn't. Like a lot of blood samples which are collected in the 60s, 70s, 80s can now be sort of looked at again and understood. So that way there is you can actually learn about long-term effects by looking backwards. We have the ability to do that to some extent, but then other things sort of, for example, how much the environment's been changing a lot since then. Like the conditions which someone lived in the 50s and 60s are not the conditions now. So how new environments might also interact with these things, you need to always keep updating that. In terms of in terms of certain things which are new and have just been found, again, it depends on technology. So for example, there's new technology in neuroscience and genetics, things like the nanopore, the Oxford nanopore, which sort of can identify a change in each gene. So each protein within a gene, the easiest way to put it. So each nucleotide or each protein within a gene, it can identify any change in a human white, which we did not have that technology before. So you can now look at those things, and then that probably give us a lot more information than what we previously did. So with the improvement of technology, we'd probably be able to do more, which we can't do right now. Yeah. So it goes in it goes in multiple ways. It's whatever you want to take it, and I guess smart people much smarter than me can do that. That's not for me to do.
Yeah, I think I've got one more question, unless Jeremy has some, before we move into the actual experiments themselves and what you found. But yeah, since you did mention that a lot of these researchers were conducted from teams based in the US, though you said some of them are recent, were there any? I can imagine, because a lot of research conducted and funded can also be based on the narrative at the time. So, were there any examples of such papers being conducted, say, during the Prohibition, where you didn't want to encourage alcohol drinking, uh, to sort of
Ooh, that's a
uh,
very good question. And that's a very good question, which I actually do not have the answer of. What I can tell you is that prohibition and then the war on drugs specifically really impacted people's ability to research, um, drugs in general. A particularly good example is use of MDMA for treating PTSD. And use of marijuana, THC and CBD. All that's grown now, but during the war on drugs, and it was illegal to do research, and very few places could actually do it. You needed to get so many permissions. There were so many hurdles which you had to jump across to actually do it. And then once you had those findings, um, because everything was banned in certain, depending on the country, you couldn't move forward very quickly or easily. So those kind of things play a role to an extent of what you could do. And I assume prohibition would have also, with nicotine's a famous example, where a lot of like tobacco companies used to pay researchers to fund projects which said nicotine's good for you. They don't do that anymore. There's a lot more stringent laws around it, around conflict of interest in terms when you're sort of publishing papers which you need to disclose. And if you don't disclose it, the papers are taken down and you lose your licenses and so on and so forth. So it's stricter now. So I hope, maybe I assume around prohibition there might have been some stuff, but I do not know. I do know of one paper from 1968 which did look at alcohol across generations, but that's after prohibition, so it doesn't really count. I think there's also one from 1920s or something, which I found, which was doing it on hamsters, moles, one of them, another furry rodent of some sort. But yeah, it's it's always a thing. Like science, to an extent, will always be political, right? What you can research, like during COVID, so much more money was put into infectious disease research, and taken away from other fields for that reason, because it was the need of the time. It really depends.
Awesome. Right. So now from there, we can just move on into the experiments and the results. So, Sahir, I ask you, what did you do? And what did you find?
I So for my PhD, what we did was we basically took a bunch of rats, bunch of male rats, and we made sure they were teenagers or young adults in terms of rat years or rat days. Because the idea behind that was that, not just idea, we did research in our lab before, and it is a lot harder for rats to start drinking alcohol, or just wild sprog dolly rats to start drinking alcohol when they're older. But if you think of it, that's kind of similar to humans, right? So like, if you don't start drinking young, very rarely do people pick up drinking later on in life. I'm not saying they don't, but I think generally speaking, most people have their first introduction to alcohol during their teenage years, and that's it goes from there. I think, Jeremy, if you have any evidence to the contrary, feel free to tell me. But I would say so, right? Because
Yeah, my evidence supports that hypothesis. Yeah.
so
Your opinion is definitely valuable here because you have spent your early years in a Western country as opposed to the other two participants in this podcast who grew up in India.
but
Yeah.
but even then, like, when if you just ask, when did you start drinking, Amer? Or when did you have like your first serious interactions with alcohol? It was
Yeah,
it was
that
around
was
like
18. It was 18.
it was like 18. Yeah, when you get to uni, you see people drinking
Yeah.
alcohol. Same thing. I feel New Zealand slightly earlier, 15, 16, maybe. Your dad gives you a beer or something. But generally speaking, people start drinking at a young around the teenage years. And it's a kind of similar thing in rats. Because what I did was I gave them like straight drinking ethanol, which means that it had no flavor. So there's no wine, there's no flavor of vodka. Rats love wine, by the way. Um, but you don't want to like that's the thing. This is why we can do this with animals, right? Because you don't want the kind of effects of the sweetness or the actual flavor to play a role in their drinking. You want just the effects of the alcohol. So what we did is we gave them like 20% pure ethanol mix in water, um, and we also gave them food, and we also gave them regular water to drink, and it was basically up to them how much they wanted to drink. And we did this for a period, and we also kind of tried to mimic human drinking a bit, where you'd give them access to alcohol for a day, then you don't give them access to alcohol for another day, and then you give it again. And this idea was sort of to kind of mimic the idea of that even humans, generally speaking, we do not drink all the time every day, right? We drink, don't drink, then we drink, then we don't, right? And sort of giving them access of whenever access so that they could drink whenever they wanted to within a period a time space was kind of this idea of trying to mimic the human experience and make it voluntary for the rats. So we weren't a lot of the research done before me has always been injecting rats with alcohol or force feeding them alcohol or giving them alcohol in vapor form so they sniff it. And so the good thing about that is you can control exactly how much alcohol a rat is drinking. We cannot control it, right? We're giving them the choice, but that's more, in my opinion, that's more kind of like transferable to humans. And because that's the goal, that's what we did. We gave 20 rats alcohol as much as they wanted to drink for 8 weeks. And then we took the animals who were the highest 10 drinkers and we mated them with female rats who had never touched alcohol or never seen alcohol before. So any alcohol in them was from any alcohol or any effects of alcohol which you would see would have been passed down from the male rats. Yeah. And then the male rats basically mated, and then the kids which came out of them out of the females, and I use male and females because rats, so easier to just delineate that way as opposed to going through genders. Is that the female rats gave birth to litters, and we sort of like did all this sort of like visual comparison. So we looked at litter sizes, we looked at the weight of each pup, so each rat pup born. We looked at like different metrics of how they developed, their eyes opening, when they started walking, when their hair started growing, when their ears started, when they could start hearing, because rats are not born with all these abilities like human babies are. They're born kind of half baked, if you'd say, where they still need to their ears still need to open, their eyes still need to open, so on. And we looked at these developmental milestones because they're usually very strict in rats. You know that on day 14 they're going to start walking. You know on day 18 they're going to start grooming themselves, things like that. And then we did a whole bunch of behavior experiments with them afterwards to see how their motor coordination is, how much they drink, how does alcohol affect them? Does it affect them differently? And so on. And then we also looked at this in one generation further. So in this case, the only interaction with alcohol they had was if their granddads drank alcohol. So we're just trying to look at whether we had the same behavior, or had the same behavior or whatever effects, if we saw any 2 generations down. And we did a lot of other experiments as well. We did one looking at their fear and anxiety responses. Their depression and anxiety responses to see if there's differences there. And then we also looked at their brains and their livers for any differences in sort of potential liver function, potential brain function. The project is actually still ongoing. After I finished my PhD, I left. I'm in the UK now, but there's new masters and PhD students trying to take a look at some other things in those animals. But yeah, that's a rough idea of what we did. I can talk about the sort of like looking at the brains and livers and the sperm and what sort of biological stuff we did, or I can talk about the behavioral stuff we did, because whatever we found so far is pretty cool. And yeah, that's a basic idea.
I'm really fascinated by the concept of mimicry in your testing. So you mentioned like giving them the ability to choose when to drink, which is something that we as like humans with agency have.
Yeah.
But then you also mentioned that you feed them pure drinking ethanol, and you're like, I don't give them wine, or you don't give them. It, first of all, like, is the mimicry to try and like sort of correlate their circumstances to our circumstances more closely, and then how do you choose which circumstances to mimic? Is there, like, do you put them in social situations as well? Because for us, right, as humans, often alcohol is consumed in a social situation. It's often frowned upon for a person to go and sit in the corner of their room and like down a bottle of whiskey. So are you putting them in like, are you giving them the free will to drink alcohol with their friends? Is it like a social thing for them, or what are you choosing to mimic and why?
Right. So this is really cool because we actually did a study. So for the purposes of my study, we did not. The male rats were isolated when they were drinking during that drinking period of this 8 weeks. Um, One of the reasons we did that is because we know isolation sort of helps with drinking. We know that from previous research and research done by our lab at the same time, um, we know that if you have a friend, your drinking pattern sort of changes in rats as well. It's the same thing. It's But we kind of wanted to see the effects of alcohol, and therefore we decided for the study that we're getting into too many layers. Um, of things if we are adding the social aspect to it as well, and then we need to control how much they drink. And if you have two rats in a cage, trying to see which rat drank alcohol from that bottle is also a thing which you need to like follow on, and we didn't have the sort of like, a lot of it is like budget and what equipment we have related as well, because we didn't have the equipment to be able to manage exactly how much each rat is drinking without completely isolating them. um we yeah oh we didn't do that. But like you said, yes, you sort of mimic, or you try to mimic um certain aspects, and then you sort of like leave out others. And the way the decision goes is, yeah, well, the one I said was the monetary decision. It just depends on what equipment you have, what you can do. But the second thing is what you're looking at, right? So we did a study in our lab at this running concurrently. Claire Bradley did that, where she was looking at whether isolated rats in this paradigm, because we're trying to mimic drinking sections, and most, like, like I said, they drink for some time, then they don't drink for some time, which is kind of like humans. And we were trying to and what she tried to do is try to mimic that exact paradigm, but have a friend rat as well. So there's two rats who are together, they've been mates, and see how they drink. And she found that drinking is reduced a little bit, which again comes to that idea of what I mentioned before, that other people have also found that isolation tends to show you tend to drink higher in isolation than you would in a group, or at least with rats. It also shows that you change drinking patterns. I think her study didn't look at it, but I think I've read papers which show that the style of drinking changes, like in the sense that you might drink more to begin with in a pair, but then reduce later on. While if you're isolated, you keep drinking throughout the day, and therefore you end up drinking a lot more by the end of the day. Um, However, uh, there's conflicting reports in terms of, like, there's multiple studies which sort of look at it, and then there's also some evidence which sort of says that, that peer group drinking, and particularly in humans, you drink more and in your groups than you would if you are alone, because of behaviors, particularly with men. I think there's like public health research which shows that, or which has been mentioned. But yeah, there's a lot of conflicting reports about it. What we do know in general is that overall, on average, you tend to, if an animal has been socially isolated, they tend to abuse drugs more. than if they're with friends. Some of the clear studies have come from cocaine research on that front, or heroin research on that front, where a rat, if left alone with a heroin thing vial, where it just like presses a lever or a button and it gets injected with heroin, it'll OD every single time. It will overdose. It will kill itself every single time. But if you have a friend rat with it, they might take some, but then over time they just stop taking the heroin, and it kind of leads to that idea of sort of like substance use. Substance abuse is sort of like, to an extent, is a disease of loneliness, is the way which it has been described as in certain papers. So yeah, does that answer everything?
Yeah, really
Yeah,
interesting.
yeah.
I think, well, so you mentioned you investigated a few different paradigms. One from, well, analyzing brain activity, behavioral activity. You've highlighted a little bit about the behavioral aspect of it. But this was during mainly the data analysis. Well, no, this is data accumulation part of the experiment. In terms of the result analysis, what sort of other behavioral components did you examine, and what did you find?
Ooh, hey, this is the fun stuff. I like this. So the findings are always fun. So this long story of about 40 minutes comes down to what we found, which is really, really cool stuff in certain aspects. So, for example, we found on average there were more pups which died in infancy in the alcohol drinking fathers versus the controls. So this is, I was suggesting sort of like maybe some effects or maybe some influence in early development. Then there were also like minor developmental delays. So I say minor because, like, suppose on average, the control rats open their eyes on day 8. The drinking rats on average open their eyes on day 9. So there's a slow delay there. These are significant in the terms of this might be showing, this kind of indicates potential developmental, overall neurological developmental delays and things. But it, on the whole, it shows that it's not that big a deal, in the sense that all of the rats would survive to adulthood, even from drinking offspring. They were behaving fine. There wasn't necessarily big behavioral changes. All these changes are subtle, which we found, which is also interesting in the sense that it kind of tells people that these things can have an effect, but they're not like, it's not that drastic that like they will die, but like these changes are sort of minimal but clear. That there might be developmental delays, there might be sort of there might be sort of growth delays. Like some rats were smaller, so they're called runts. So alcohol drinking offspring had smaller runts. That kind of links back to what I said in the beginning about IGF2, as that's one of the genes which is affected with alcohol and it impacts growth. So there could be linkages there. So that was development side. Then when you go on to behavior, this is where it gets really interesting. It looked like we found sort of differences between the female and male offspring of fathers who drank. So you sort of see this difference. The most interesting one was in how sensitive they are to alcohol. So one really a really funny experiment which you do, what funny was it really funny for you guys, is you know how like cops sort of ask you once you're drunk to like walk in a straight line to see whether you can, right? It's sort of the same idea. So you put rats on a sort of treadmill which is raised high above the ground, and you sort of see how long they can stay on it, and then you inject rats with a dose of alcohol and see whether they can still stay on it, and then you inject them with a higher dose and see if they can still stay on it. And the performance drop is generally very consistent. So if the higher dose of alcohol, the quicker the performance, your performance drops further, right? So if you're given 1 gram of alcohol, you say if you can stay on the rod for like a minute, you're given a gram of alcohol, you stay on it, gram per kilogram of alcohol, you stay on it for 30 seconds, you're given 2 grams per kilogram of alcohol, which is double the dose. you stay on it for like 5 seconds, just to give you an idea. And what we found was controls had like a nice, neat sort of decline. But the offspring rats, you find that particularly the females across the first and second generation, that first dose does not seem to impact them as much. They perform their drop in performance is nowhere near as steep as the offspring of control rats. So sort of like this change in sensitivity. The other thing which we find was, remember I told you guys about, we just gave them alcohol to drink. We gave them a choice. We did the same thing in the offspring. We gave them a choice, and the male rats drank as much as the control males did. So they drank almost the identical patterns. But the female rats drank a lot less than what their counterpart control females drank. So sort of this idea that there's something going on there which might impact the way female offspring of male drinkers might be interacting with alcohol compared to male drinkers. So yeah, those were two of the really cool things which we found behaviorally.
Yeah. Wow. So here's that's
Yeah.
very
Wow.
yeah. That's that's very fascinating. But here's sort of, this is a sort of a segue question, but it is from the wrinkles I gained listening to your listening to your episodes on statistics. So if you are not happy with the question, my only response would be you reap what you sow. What was the
Get
uh,
'em.
what was the sample size for each generation, and how would that affect the overall findings in terms of translating it to the real world?
Right. Yeah, so animal research always has small samples. Okay. One is because we don't want to hurt more animals than we need to. Two, it's to give an idea. It's not what we find is generally to give ideas. Like all these things you have said gives us an indication of what might happen, what might be happening. But at the end of the day, all these things need to be done in humans, and that's where we go for the large sample sizes. Um, there's things which we mitigate, right, to allow for stronger statistical power. So for example, all these rats have almost the exact same genetic makeup, right? So we use a particular strain of rat. I used Sprogdolly rats. My previous experiments, which we had done, we used Wistar rats, but we use a particular strain of rat with almost identical genetic makeups, so that there's no sort of genetic differences which you would see within humans. In humans, you need large sample sizes to kind of control for that difference or that variance. If you don't have that, that's one thing which you can control for, which allows for a smaller sample size. The other thing is genetics. The genetic variance is one thing, but then there's also environmental variance, right? Like the way Jeremy grew up is very different to the way Amer grew up. The way Amer grew up is also still different to the way I grew up because he's a younger brother. So the experiences which he faced would have been different to me, and you have to account for all that variance. Over here, the rats have been grown up in the exact same environment, exact same temperature conditions, exact same sort of cage sizes, exact same amount of time away drinking, exact same. So when we're controlling for all these things, it allows for smaller sample sizes. Having said that, we made sure that none of the rats were siblings in this experiment in terms of so for example, the 20 rats which drank alcohol, none of them were at the start. So the fathers, none of them were siblings. And also, although they have the same genetic makeup, they didn't have the same mother. So there was a little bit of variance to see whether this still holds, because you don't want the case that it's just one same rat family doing all the experiments, and then the problem might only be with that rat family. Yeah, if I do the experiments from from one clan up in Scotland, like the McDonald clan or something, it's very hard to translate it because they would
I
still be like
believe
isolated enough.
to our listeners, I believe that would replicate an incestuous environment.
Yes, exactly. We don't want it to be too incestuous as well. It's incestuous enough that they're all the same genetic makeup, so there's not much variance that way, but you don't want it to be that it's causing other greater problems. And then when I talk about the other experiments which we did, the alcohol sensitivity one, the alcohol drinking one, the behavior anxiety test, the development, the ones whose brains we took out, all of those were done on one animal from each litter. So each pup litter gives about 10 animals roughly. On average, 5 male, 5 female, but it's not always the case. But yeah, on average, we're talking about 10 to 12. And we made sure that there was one male and one female from each litter in each experiment, so that there's no sort of overlap between experiments and there's no sort of multiple siblings in one experiment. So that's so sample sizes. If you want sample sizes, there were 20 in the first, 20 fathers, and then after that we're talking about there were nine drinkers, non-drinkers in the first generation, and 8 drinkers, 8 non-drinkers in the second, because in each case, like one father did not produce offspring. So in terms of animal research, it is on the larger side actually, because most animal papers which you see, particularly like the bio papers, usually only use 4 or 5 rats. So using 10 is larger. So that way it's good. But yeah, you're right that sample sizing, as a statistics person, you would say that you'd need a larger number, but we're sort of countering that with trying to make conditions as similar as possible to control for all the possible variants. Yeah.
Right. And now I have a question for Jeremy. I think we've got some time to examine an additional aspect of your findings. So Sahir mentioned he's looked at some neurological activity, genetic activity, examined the sperm. Which of these aspects do you think he should delve into?
You're asking me.
Yeah.
That's a crazy, crazy question. Actually, well, can I answer your question with a question?
Yep.
Because I was going to ask you, Sahir, and I think this is where I'm really interested in talking to you in the academia circles that sort of surround you, is like I always feel like a bit of a dummy. And the thing that I always want to know, and that we've talked about in your research and some other people's research, is like, how does this manifest for us, like as a society, and like how do you hope that it will manifest? And I guess that will answer Amer's question as to where I think you should go, because I think you should go wherever this will best benefit society in the future. Like, how do you want to see it manifest, I guess?
Oh, like my research, like the output of my research. So I would like to see it manifested on like the research side. I would like to see it manifested in people doing this work on humans, because that's the goal, right? We want to make sure that we're having healthy babies, we're able to take care of like our society moving forward. And if drinking alcohol before birth is showing these effects, which it is, then we should see whether this translates really well in humans. There's some evidence that it does, but we need more work to sort of quantify it. And then on a wider scale, you want like this to influence policy, so sort of policy work around this. So people plan like family planning and so on and so forth, for example, would be a good way to think about it, where you'd like to see it manifested. The other place where I'd like to see it manifested is being spoken about, because talked about more maybe in the media space a little bit, in the sense that we associate with drinking and smoking being cool, right? Like in media, like James Bond's all about sipping martinis. It's the whole idea of drinking and partying is associated with being cool, which it probably is to some extent, right? Like people enjoy it. Like alcohol is a drug at the end of the day, which you enjoy. But then also sort of being aware of what excessive drinking could potentially do in terms of not just to you, but also potential kids you're planning to have, potential future effects. I think that's where I want to go with my research in general. But I think it's also there with science, right? So science inherently is all about you sort of asking questions and being like, you can never prove anything really in science. You're always disproving things, right? So you're always saying that no, this is not what happens. We cannot guarantee tell you that this is exactly what happens, but we're 99% sure or 95% sure that this is what happens for whatever hypothesis, like XY. And what that means is a lot of research, when we speak about it, comes with that uncertainty because science in general is uncertain, and which means it makes it very hard to sort of communicate when we get into the wider discussion of science communication, which allows for a big room for misinformation, because people don't like to deal in like maybes and is it possible and all that stuff. You want to know, okay, is this bad for me or is this good for me? And a scientist can't go, well, maybe under these circumstances it's bad for you. Like, and that's where we're at with science, right? And I think I would like to see people like, well, you, Jeremy, because you're a creative person. I want people like you, sort of like putting, giving ear to to like science research a bit more, but also understanding how we can potentially show or send these messages to like wide audiences, to the public, to kind of understand like the basic premise of science and what things, and if you find a finding, what things to be wary of. Not everything's good, not everything's bad. Like you see it a lot with marijuana research in general, or being like, there's people who are like, no, it's really good for everything, and then there's people like, oh my god, it's terribly bad, when the truth is like there's good and bad to it, and you need to understand and be educated enough about it. So yeah, I think that's where that's where I'd hope it to go. That's where I want it to go. We'll see if it does. But yeah.
That's a good point. The middle ground does not sell, and it's something that you see really predominantly in Hollywood. Something that you see in a lot of the like entertainment media which we consume, which a lot of our like social and popular culture revolves around, is like good and bad. The concept of like, this is good, this is bad. There's not a lot of room for the public perception to be like, this is good in some circumstances and bad in others, and there's a whole lot of nuance that goes with it. I think you're right. There's a challenge there in our world and in your world to sort of come together and begin communicating that more effectively and a little bit more connectively, so that people can understand these things and hopefully end up better off for it, right?
Yeah, I think the other problem is that we also need to realize that the middle ground doesn't necessarily mean that it's in the middle. Like it's
Yeah.
not that like it's good, it's bad, and the middle is just like, oh, it's nothing. You need to sort of understand that that's also not true, depending on what we're talking about. Like we can be like, it's not like with the flat earthers and round earthers that, oh, the earth might be a triangle. It doesn't work that way. It's not that middle ground. But there is that sort of thing of, particularly in psychology research, because you're working with such vague terms in many ways. You're working with behaviors, you're working with emotions, you're working with sort of things which are slightly nebulous. Qualitative research makes it even harder because you're working with lived experience of people and you're trying to amalgamate it. And yeah, it makes it really, really hard, and people need to be able to understand the nuance, and we need to be able to open spaces where we can have that conversation with that nuance of like what all is good and what sort of bads we need to be worried about.
Sort of just goes into that aspect of science being in publications being a peer-reviewed process, right? You kind of want it to be true to that aspect of, yep, there is a lot of gray area, but within that gray area, what what has the person investigator find, and what have the peers cross examined, and being able to collectively move science forward rather than just finding loopholes within your own research to try and find a revolutionary hypothesis statement which can be easily challenged, but at the same time is not really challenged, because those taglines are what are attractive to the mainstream. it's just something that feels even more lost in this sort of era, where you're trying to just push forward a narrative rather than trying to trying to gauge. Okay, you're trying to have that being willing to have that discussion in the gray area.
This is
Yeah.
a huge crossover area between our worlds.
Yeah.
Headlines being like a major issue.
Yeah.
Everybody likes examining things on the surface level because it's easier to see and it's easy to observe and it's easy to quantify and articulate, right? So it's easy for them to explain to their peers and it's easy for them to sort of bring it into their own worldview. And correct me if I'm wrong, but like the psychology of humans boils down to like, we'll just do whatever's easiest a lot of the time. Like,
Oh yeah. Um, I've wanted a proposal of, because in journal articles the peer review thing is you have other like I wouldn't say fancy scientists because I'm technically now a fancy scientist who does peer review stuff as well, which is shocking how they let me do it. But I feel one of the things which you need to uh which we need to add to peer review is like, how like jury duty works in most countries. So for example, people in Europe where they have a sane judicial system, it's not for you guys, for the French judicial system, it's for people in the UK, USA, New Zealand, Australia, where there's a jury system where you call random people from the public to decide on whether someone's a witch or not. And I feel it's you should not do that in law. I don't think, at least personally, I don't think you should do that in law. But you probably should do that in peer review, where you should just send a journal article to an absolute random person and be like, do you understand this? Because if you understand what the guy is trying to say, or like a version of the article, you could keep the hi-fi version for the academics to understand. But you need to send it to like some random member of the general public, and if they can understand what you said, then and only then it should be published, because you should have a clear understand. You should be able to sort of manage expectations in that way and sort of cater. But I don't think that's possible viable. It's just like a favorite dream of mine to move academia forward, because at the moment half the time I read articles and even I'm like, what the hell are they talking about? It feels like I needed a thesaurus, but then I felt the thesaurus had too many words about a dictionary, and then worked my way back up. It's the same.
That's a common stereotype of uh people in STEM, right? That their communication, especially written communication, unless they're in psychology, is absolutely horrendous, and it's a stereotype, but it is
Oh,
fairly prevalent. That
some of the
yeah,
bio papers, some of the genetics papers. Oh my god, it's they're speaking in tongues. But
funnily enough, Sahir, you've brought us full circle in a sense because you've highlighted this need for the general layman or general public to understand these quite complicated academic sort of pursuits. And we've had a lot of discussions about this because I am one such person, right? And I think this project and this podcast in particular, the Smooth Brain Society, is in at least ethos and in practice now addressing that gap, and is sort of like more and more moving towards addressing that gap more effectively. So it's kind of like we've just been talking about your career thus far, and now this element of it is, it's almost a self-fulfilling prophecy. And do you think that is why you started this podcast?
yes, yes, time to plug the podcast on my podcast. How good.
this was the intent all along, wasn't it?
You've done this entirely yourself. You've come this full circle on your own, which is amazing. I'm literally sitting here going like, hey, does he see this connection that he's established for himself?
No, not at all. Damn, you accidentally fall into a lot of things, do you? And yeah, that's I guess to an extent, yes. It was my goal with the podcast to some extent. I remember speaking to you even before I started the podcast about wanting to do something like this for a while. Amer would remember with our flatmate Henry, who did psychedelic work, that we wanted to do this podcast, and we did a pilot episode which should never be aired, because it went down too many terrible rabbit holes.
I
But
believe I was the host in that one.
you were. So look,
Yeah.
the host of the very first unofficial episode is now the host again. Only took you 47, well, technically 50 recorded episodes to come back. Yeah. Any anyway, so it came from the discussion where we were making, as you know, Jeremy, and most of the listeners, but I'll repeat it. The idea of having a smooth brain is like not being very smart, and the idea of a wrinkly brain, because brains have more grooves, and the more grooves you have technically means more surface area, which means you should be smarter, right? So we were talking to some people about their work, and they were explaining it in a way too fancy manner. And one of our other friends, Joe Corn, said, "My brain's too smooth for this. You need to dumb it down." And that's kind of when the idea got picked up, and then that was later in the evening is when we sort of recorded the first episode, which is never going to air. But it was basically based on that idea that we are not smart, we don't understand this. Which is funny enough, the person who was explaining the work is another person in psychology, and it's just a different area of psychology which we had no clue about. So it also shows that even within these fields and all that, there's specific things which we can be really smart in one thing. Like I know quite a bit about rat work in alcohol, but I would be completely lost outside in a different field, and I have no clue, and I'm just as smooth brained as everybody else about it, apart from somebody who works in that area. And that's where the idea came from. Then a few of my students, because I was tutoring, they kind of were scared of our lecturers, or were sort of intimidated of them. And because by this point I was a PhD student, and I knew that all of these lecturers were also softies and sweet people, and they also have their own like flaws and good points and stuff, I thought that I would interview them and sort of put them out for, the other psychology students, other undergrads, to feel comfortable that, these guys are humans too. And that's where the podcast was kind of born, where in terms of its current format, where I decided to get someone on who has no real clue about the subject. Most of the time I have no clue about it as well, and then we talk to somebody who really knows their stuff, and sort of we learn together. And that's basically the full circle plug. Also, new plug, we just started our Patreon, so we've been doing it free.
Hey, I'm the host. I
Bye,
should be
bye,
saying
bye,
that.
bye.
I should be saying that. Well, the Spoonbird Society has recently started a Patreon, and we thank you. We've also received one. We've also received one patron very recently. So thank you to that person whose name I do not know because I am the interim host. And also would
Garrett. Thank
encourage
you, Garrett.
our listeners to explore the Patreon, where you could potentially in the future, given the demand, also provide some outtakes and extended cuts on some of the discussions that have nothing to do with science, and just general banter.
The fun bits.
Yeah.
The nice cool fun bits.
Yeah, exactly. Because the whole purpose of this is, again, to give science a platform, give everybody access to it. So we don't want to put things behind paywalls. That's why I've also sort of like working through with Europe's Journal of Psychology and PsychOpen, which are publishers which publish research open access for everybody to read. And that's the same thing with this. We don't want to put anything behind a paywall. But yeah, well, of course, this needs to be funded. We've been doing it on our own. So any support is really helpful. And if we could do extended bits based on demand, sort of like, non-science chit chat, maybe even ask questions, get researchers back on to sort of answer those. Some things, let's see what we can do. It depends on what we have the resources for.
Yeah,
But
that's
great
a great
plug.
idea. Yeah, that's a great
That
idea.
sounds like good value.
Yeah.
Wow. That sounds like incredible value.
Definitely does. If I weren't the host, I would donate.
If I were not the co-host, I would also donate. If I was an unbiased third party, I would be clicking that Patreon site. That's www.patreon/ I'm assuming Smooth Brain Society.
It is Smooth Brain Society.
Hell yeah, I just guessed that. Go,
Yeah, easy.
go
It's
and
that
donate
easy.
just because I guessed.
Yes, it's that easy, our listeners. It's genuinely that easy.
You can also find it through our website at smoothbrainsociety.com. So yeah,
Do that too.
So-
we got our URL sorted.
Yeah.
I would like to say that I think this is and Sahir, you know this, I think this is such a cool concept. I learn so much every time I listen to one of these. I learn even more when I've been on them. And it always feels a little bit like deep end. And I'll never forget. I'm about to name drop a few people, but I'll never forget being in a room with you, Sahir, and a lot of your friends who I was meeting for the first time. So Ali and Bailey and Finn and Nusha and that group of people. And I was sitting at this table going I am so dumb. I am so dumb. But the minute that you start talking to those guys about anything, you realize that it is not their intent at all to make you feel dumb, and they are mortified to know that you would be feeling that way. So I think this as a platform is such an awesome facilitator to break down that boundary and to let people understand that the world of academia is full of really, really smart, intelligent people, but they are also people. And the root of connection is that people to people conversation.
Yeah. No. And like you said, to be fair, that same group, that same room, we think about how smart and creative you are in your field because we have no clue about this when you just show up with your artwork. So Jeremy does a lot of sketches, does a lot of production, digital work, worked for Pickpock as a designer, was creator, designer. What else did you do? Producer of
So I the cinematic director.
Cinematic director. There we go. Not designer, director. There we go. But you see his artwork. You see his CV. His CV went viral about 2 years ago because he sketched the whole thing. And when we sort of see him, we're thinking probably the same way he was scared about us because it's just not our domain, and we need to understand that everybody has something to share and everybody can, and you can learn from anyone as well. So yeah.
Yeah. Well,
that's what it's all about.
Yeah,
Good job.
Exactly.
It is.
Yeah. So for our listeners, if you were wondering if our regular host, Sahir, had a smooth brain or a wrinkly brain, he clearly had some predefined wrinkles before he started this podcast, as indicated by his knowledge on alcoholism displayed during the course of this episode. So there you go, in case you were wondering. But yeah, naturally, I think we should wrap up there. Yep.
Yeah,
Thank you so much for listening. I have been your host, Amer Hussain, for the first time, and maybe not the last time. We'll see. In case Sahir wants a follow up of this episode. Thank you to Jeremy.
Maybe I can
us.
outsource my hosting duties
Thank
to you.
you guys.
Yeah.
Oh, that was really fun.
Yeah. See you next time.
Awesome.
What's the sign off? Do we do a sign off dance? Is there you got to do a dance on TikTok now to get things going
I got to
viral.
start it, and then it's got to go. Who's in the middle? Sahir's in the middle.
Sahir's in the middle. Yeah.
No, wrong side. You got to start the other way. Yeah. Yeah. There you go.
Okay. Oh.
Yeah.
Yeah.
Hey.
There we go. Got it.
Yeah.
Awesome.
Cool.
Hey, you didn't ask for advice from me, but you don't need advice from me, so it's all good. Thank you so much.
I always.
Oh yeah, sorry. Oh yeah, I forgot about that. Oh, if you had, I think the podcast itself, listening to the podcast would be the advice you give to people. Let's be honest. But if you had one piece of advice for our viewers, not viewers. Sorry. You're gonna have to cut that. If you had one piece of advice for our listeners today, Sahir, what would it be?
I asked the question and then I had no actual advice. No. I've actually got a lot of I've got a lot of advice over the course of my life, really. Apart from all the advice given by all the podcast guests before. Do not take a Snapchat saying about to be randomly selected before getting through security at an airport. You will always get randomly selected for that. Do not take pictures on tops of cliffs, trees, etc. Does not end well. Just generally, someone had once said, "Be better," and that's always a good one. Be better. Don't be a coward.
Sahir always uses those on me. Be better, don't be a coward.
Nah, nah. In general, just everybody take care of yourselves. Take care of your mental health. Keep having these conversations and subscribe to the podcast. Yeah.
Nice.